Good evening, ma'am.
Sir, you can start, sir.
Yes, ma'am. Thank you. Good evening, my dear students. You are all at warm welcome to the once again the biological class. Last class, what we have discussed already, about. So one is called as what are the types of chromosome? Artificial chromosome is the bacterial artificial chromosome. It's simply called as BAC. Yeast artificial chromosome YAC, and mammalian artificial chromosome is called as MAC. Already we discussed. And then, so vector, what are the types of vector is there? So already about plasmid, bacteriophage, plasmids, and PAC, YAC, and so and so. The size of the molecules, the inserted size, how much is that? That things also already we discussed. Which one is the lowest one? Which one is the highest kilo base inserted size? It will be depends upon the vectors. Okay, they may be differ. So already we discussed. Next, we are going to discuss about the important part. Okay, so this is also a very, very important, just like a DNA primer, RNA primer, where we were used, where we will use these things, maximum RNA primer mostly used for the transcription or replication process, isn't it? DNA primer is also where used, where, the PCR polymerase chain reactions. Okay. Likewise, the marker gene is also very, very important, play a role in which one, genetic engineering and biotechnology. Okay. See, so what is by marker? It is highlight the gene sequences, isn't it? Marker mostly used for highlights of the gene. Here, the monitoring and the detections of the plant transformation system in order to know whether the DNA has been successfully transferred into a recipient. How do you know whether gene is transferred? Right, okay. That gene is whether it successfully reached the target cell or not. How will you know? We do not know. So that time they were used a specific genes for what is the identification of the transfer genes. Such type of genes are called as what is known marker genes. Such type of genes are called as what is that? Marker genes. See that DNA has been successfully transferred into the recipient cell is done with the help of a set of genes. This set of gene is called as what is the marker gene. The set of gene is called as what is that? Marker gene. Don't forget, my dear. Okay. So it is used for the which purpose? With the DNA donor as well as a recipient. Okay. In between, it play a vital role. That is very, very important. Okay. Go to the next one. The marker genes are introduced into the vector along with the target gene. This is the point. So in between the, which one the vector and the target gene, in between there is a mediator is there. Mediator means it is a essential role played by the marker genes. Okay, that is very, very important, my dear students. The marker gene is or introduced into a vector along with the target gene. Okay, so this is the marker gene. See, this is considered as a plasmid. Okay, this plasmid it contains various gene sequences. Already know, but there are different colors have been reported here. Particularly, which one there is a blue color, light blue color have been given here in this part. Okay, this part is what is that target gene or gene of interest. This gene is interested to what is a hybrid with the another one, otherwise called as gene transformation matic genes, like that we may tell or told. Okay, this part. Okay, so then another one is the selectable marker. See, this is called a selectable marker. So this portion is called as what is a selectable marker. Suppose if it is attached with the target gene, we can identify this one is called as what is that? Donor. Okay. Suppose if it is joined with the recipient cell. Okay. Recipient cell means along with the marker. So this one is donor genes. Remaining things are recipient genes. We can easily identify. There is a small marker. The marker is also one of the genes fragments, otherwise called as gene sequences also there. And the specific marker is also having in commercially also there are so many markers also present in it. Selectable marker, they are all are genes, DNA molecule, a fragment of DNA molecule present in it. Okay, they are collectively called as selectable marker. Okay, each one it has each plasmid, vector, bacteriophage, plasmid, and cosmid. All those things they are having a specific recipient marker also. Okay, each genes they will not accept all the markers. Okay, that is the reasons. See the next slide. Marker genes are categorized into two types. The marker genes also further divided into two types. So one is called as reporter genes, another one is called as a selectable marker. Okay, so it may be divided into two according to their usage. Okay, the reporter genes and another one is called a selectable marker. Okay, so first one is we are going to see that reporter genes. What is by reporter genes? See here a number of screenable or scoreable or reporter genes. The reporter genes also called screenable or scoreable or reporter genes are available that show immediate expression in the cell or tissue. Such type of immediately you will get the report. What is the meaning of report? Report is nothing but the action of the genes. When you are introducing inside, what happens, immediately it will express in themselves. Okay, see that one. Reporter genes. A number of screenable and scoreable of reporter genes are available that show immediate expression in the cell and tissues. A reporter gene is a test gene whose expression result is in quantifiable phenotype. That is very, very important. Quantifiable. Quantity means how much quantity is there. It has a specific quantity. Suppose it will not enter in much more. Okay. So that is very, very important. Reported genes and what will be the percentage, how it will be expressed, in phenotypically express itself. That is very, very important, my dear. See the structures. This is the plasmids already in our board. Already we discussed a previous slide. This gene is the violet color, purple colors is indicates the a gene of interest. Already we know what is that? Ampicillin resistance or whatever it may be, tough, cold, and anything is whatever it may be. In this portion, see that this is the red color. There is a selectable marker. Okay, it's included. This is the entire plasmid structure. Each one it has a specific structure. BP 322 and also 25, 27 like that, and EP, IEPM. Okay, that things also. And second one is called a selectable marker. Okay, selectable marker. What is the meaning of selectable marker? You know very well about that. What is meant by selectable? Selectable means the heading also having the answer, isn't it? Selectable marker. The selection of transformed cell is the key factor in developing the successful method for the genetic transformation. Selection of transformed cells is a key factor. Which one is the key factor? The selection of transformed cell is a key factor. Okay, this is done by the certain selectable marker genes that are present in the vector. So along with the gene of interest, this is the important. Present in the vector also. Already you know all about this is the vector, isn't it? This is a plasmid. Already you know all about. In these plasmids, this is the interested gene, right? Okay, already you know all about. This part is this is a selectable marker. Okay, already present in the vector only. Okay, this particular genes also encoded particular sequences. Okay, say that. This is done by certain selectable marker genes and that are present in the vector along with the gene of interest. The selectable marker are an integral part of the plant transformation strategy. Selectable marker is also very, very important. Okay, and also it an integral part of the plant transformation strategy when you are going to take any two plant. Okay, and the plasmids also isolated. After that, what happened? The gene transformation take place within that. During that time, what happened? The marker is very, very important because both the plasmids are same in nature, the colorless and also invisible in nature. Okay, how will you identify it? Which one is the donor? Which one is the recipient? See that one, then only it will be better for your understanding. Already we saw it. See this one. Take an examples. This is the A. This is the A plant. Already you know about. This is the B plant. Okay, already we saw it. All those things. This is the A and this is the B plant. So the plant leaf structure is the okay. So already you know about that things also. Here I have given different colors. Then only it will be better for your understanding. Okay. Okay. This one is called as A sample or plant. A stain. This one is called as B. Each one it has a specific cell structure. Already we know all about. Already we saw it. All those things. Anyhow, today also we have to discuss this part. Okay, this is the A part, and also this one is a cell. This one is also isolated from the B cell. Okay, from this part, any part, we can make a cut and also isolate like this, and here also the same portion. So okay, remove like this. So then what happened? See inside already but there is a plasmid. Is that plasmid is a extra chromosomal body present in it, isn't it? extra chromosomal body, and also the plasmid is also present in it. At the same time, what is that? You can choose the another part. This is considered as a extra chromosomal body that is called as plasmid. Plasmid. Okay. Both are plasmids, isn't it? So each one it has a specific name also. So isolate outside. The structure will be like this. This one is considered a B strain plasmids. Okay. The next one is, this is considered A strain plasmids. This one is A. This one is B. Okay, this part will be comes out. This is also comes out. Okay. Next, each one it has a specific marker gene along with the what is a interesting gene also present in it. Okay. Take an example. So in this part, so okay, in this part, so there is a marker gene in this part. So this is the marker gene. Marker. Okay. This one is called as marker and gene. Okay. Likewise, here also there is a marker gene. Here also there is a marker gene. This one is a marker gene. Simply I tell you MG. Okay, marker gene. Okay, but both are come and contact close to the how it will be all the things, all the plasmids also cut with the help of the molecular scissor. Already know about restriction endonucleases also there. You have to make a cut. Okay, cut after that. Along with it, this one is this portion. So take an example. This portion is a interesting gene. Interesting. Okay, interested gene. Okay, interested. Okay, this part is interested gene. Likewise, here also you can give you different colors. It's okay. Different colors means you can easily identify. Okay, so this one is the interested gene. This portion, just imagine this portion is interested gene. Here, this portion is interested. Okay, so here the marker gene is also here. Here the interested gene is also there. So both are, what is that? Both are common contact close to that. That condition is called as hybridization. Hybrid is combination of two different plasmids. That is called as hybridizations. Okay. So here I have given is the different colors. This is the red color. This is the violet color. Otherwise called as pink color, purple color, whatever it may be. Okay. But in plant cells, the plasmids are colorless in nature. So that time what happened, we cannot be able to find out which one is the donor cell, which one is the recipient cell. Both are common contact close together with the help of the technique is called as hybridization technique, otherwise called as protoplasmic fusion. Okay. During that, it's also after the combination of these two genes along with the markers. Okay. It will be introduced into the which one host cell. Already that that things is also already known. Okay. This one, so this one, okay, already know like this. Okay, have to add this here. So red color. Okay, you have to choose the red color and also add here. So this portion is called as what is it? Okay. So along with the marker gene also. Marker gene means which color is there? It is just imagine this is considered a black one. Okay, just okay. Here there is a small marker gene is there. Okay. So now the replication takes place within the coast. This is the coast. Okay, within the coast. Already know about. This is the coast. Maximum E. coli. Already know about. E. coli is proper. E. coli is one of the bacteria, and also it is also one of the vector used mostly. See, once again repeat for you. But this is the one of the technique. Okay. What of the technique? That is nothing but the protoplasmic fusion. The plants also selected A plant and B plant. Suppose take an example. This one is called as a tomato plant. Tomato. Okay. Tomato. Okay. So take an example. This one is called as potato. Potato. Okay. Potato plant. Okay. The potato. Okay. Each one it has a specific properties. In our tomato, the color, fruits will be, where is it? Where is it? In the upper part, isn't it? Upper part, you will get it. Okay. The fruits. Fruits is there. Okay. Here, the fruits you will obtain, isn't it? Fruit is there. Okay. And potato, what happened in the ground part, don't forget. Okay, the ground part, what happened, there is a potato is there, isn't it? The potato, yeah, there is a small eyelids also. Okay, now we select these characteristics. Which one? The potato as well as the tomato. The plasmids also, it has a specific characteristics responsible for the fruits, responsible for the vegetables, underground stem. So both are united together, okay, during the process hybrid. It is also introduced into the host cell. As a result, what happened? Both the genes along with the marker genes, that is very, very important. Now, we may consider, we may come to the conclusion also. How? Which one? We cannot able to identify here. Which one is the donor? Which one is the recipient? Okay. This one is called as donor, isn't it? This one is called as donor. Okay. This one is also a donor, isn't it? Sorry, this one is recipient. Sorry, this one is a recipient. Okay, recipient. So this one is called as donor because it is isolated from this part and marker also. This only. When we introduce the two things is there. Okay, selectable marker. In this marker is also we may add here. So this is also a B gene. Take an example. This part is B. This part is A. This one is called as host. So total 2 gene of these two combination present within the host. What happened? The another plant. Okay, another okay. See here. Okay. Okay, here. So the both the genes, okay, how it will be the result? What will be the result? They undergoes the replication process after the introduction of these two genes within the host. What happens? The undergoes the replication process. It will get, what is that? Replication of both the genes. So what happened here? So both the plants character will be pure. See that one? See this one. This is a okay. This is a E coli. That is called as host. Already we introduced which one? Both the gene within the host, isn't it? Suppose take an example. This is the host DNA as well as the plasmid. This is considered as a plasmid. Already we told. I simply marked it. This one is called as donor of A. Okay, and another part. This one is called as B. This part will be B. Okay, this is host gene along with the marker. Okay, this one is called as marker. Okay, so now after that replication process, we will obtain the 3 gene sequence, isn't it? After the application of these things, what happened? The replication enter inside the host cell. You will get these structures, isn't it? It introduced into the which one? The plant. It introduced to the plant, isn't it? Introduced into the plant. Okay. Introduced into the plant. What happened? The new plant. This is the new plant. This is the new plant. Okay. The new plant, it has both the characters of which one? the new plants. New plants. What are the characters present in it? Both the plant character will be appear here. How it will be? No, the lower part already we discussed. Lower part there is a potato is there. Potato is also we'll get it. Okay, in the upper part what happened? No, there is a fruit is there. Upper part there is a fruit is there obtained. So this is the beneficial. The upper part is also potato. Sorry, tomato. Okay, tomato. Okay, tomato. The lower part, what is that? Potato. Okay, potato. Because these two specific interested gene will be, these two genes are isolated from the specific plant, two different plants, isn't it? These two are two different plants. Plasmids are isolated and also undergo the hybridization process. After the hybridization, what will happen? You will get the new variety. This is called as a new variety. In this plant, the upper part they will produce which one? Tomato, and lower part it will produce potato. So two beneficial or bi-beneficial in this plant. This is the technology. This is the genetic engineering modified technology we are used for. What is that? Commercial purpose. Okay, so that is very, very important, my dear student. Don't forget. Okay, this is very, very important things. Okay, this one. See that the selectable marker are the integral part of the plant transformation strategy. Plant transformation strategy. Selectable marker genes enable the transfer cells to survive on media containing toxic level of selection agents, whereas non-transformed cell get killed. So this is very, very selectable marker. Also, sometimes they may be survive. They will not survive because sometimes what happens, the culture medium. Okay, which one in the media? Survive on media, isn't it? The survive of media. Maximum what happened? The agar. Mostly they were used. Sometimes they were used. Suppose if it is a blood culture. Okay. Suppose if it is blood culture means what is that? Sometimes it may be a toxic substance present in it. What happened? It will not transfer. So the toxic substance killed the gene. So that time what happened? You will not get the proper result. There is a reason. So the culture media is also very, very pure in nature. It is help for the development of the cells or that hybridization technique and so on. So suppose if it is not interfere with the negative result, it will not give any negative result. That is also very, very important, my dear. Okay, go to the next one. What are the selectable marker? What are the difference between selectable marker and also reporter marker? See that one. There are 2 or 4 points have been given here. A selectable marker, a gene whose expression allows one to identify cells that have been transferred or transfected with a vector containing a marker genes. Selectable marker, normally vector with the marker gene is also very, very important. Reporter markers, they are gene used to tag another gene or DNA sequences. Tag means just you are going to type like that. Okay. And second things, the selectable marker help to distinguish between the transformant and non-transformant. We can easily identify it. Helps to distinguish. Selectable marker means already we know the concept. We know the content of the gene. We know the nitrogen basis of the gene. Selectable marker, it has a specific sequences. That sequence already known one. So we can introduce into the course cell what happening. It can easily identify which one is a transformant, which one is non-transformant. Like that we can easily identify. But in this category, in reporter gene help to measure the amount of expression of transformed gene one day. Reporter genes, the amount, the quantity will be analyzed. Okay, analyze and express of transformed genes. How much amount of transformed genes we can introduce into the course that can be easily identified with the help of the reporter marker. And then the selectable marker has its own promoter. Okay, selectable marker is also, it has a own promoter. It is also very, very important, my dear students. Sometimes they may ask the questions, selectable marker, it has a own specific promoter or not. Sometimes assessment reasoning. Okay. Okay. This is very, very important. and regulate under the promoter of the transformed genes. This is also another one, regulated under the promoter of the transformed genes only. Example, antibiotic resistance gene, herbicide resistant genes here in this part. Sometimes they may ask the question, selectable marker. So what are the examples? Antibiotic resistance, take an example, tetracycline, ampicillin resistance. Already we studied in the plasmids, BP322 and so on. So in this category, there is a GFP and also luciferase. It's nothing but the enzyme. This is luciferase. It's a reporter marker. These are the differences between these two. Okay, go to the next one. Marker genes help in identifying and eliminating non-transformant and selectively permitting the growth of the transformant. This is very, very important. The marker, what is the main help? It can be easily identify the non-transformant. Suppose if it is not non-transformant gene also present in it, it can be easily removed. Okay. And also at the same time, which one is transformant gene? It can be easily grow within the host. Such work done by which one? The marker gene. That is very, very important. Okay. Next, in order to that foreign DNA can be cloned into the plasmid. Foreign DNA means vector DNA. The plasmid should possess only one cleavage site for at least one restriction enzyme, and this should be in a non-essential organ region. Okay. So non-essential region is otherwise called as what is non-essential. Already we discussed one enzyme, one cleavage site, isn't it? Already we discussed in the previous sessions. Okay. What is the use? There is no use of that one. So it can be removed. It's better for our practical oriented, otherwise called as procedure. Then it will be successful. Otherwise, what happened? It will not success. So the marker gene, what happened? It is essential for the identification of eliminating particularly non-transformant genes, and it is essential for the growth of the transformant genes. Okay, the non-essential region is also has what is that? One cleavage site and one restriction enzymes. Such genes are called as what is that? Non-essential region, or otherwise called as non-essential genes. Okay, this is very, very important. Go to the next one is, see this one, the antibiotic resistant genes and their mode of mechanisms. Already we studied so many antibiotic resistant genes. Okay, here they have given is a 4 only. The ampicillin already we studied. Kanamycin is also another type of antibiotics. Tetracycline. Okay, this is a important one. So this is also a very, very important one. Tetracycline and chloromopenicol. So these are the things. These are the antibiotics. Which gene is responsible for this one? Gene product is a beta lactamase. Okay, beta lactamase. Don't forget, my dear. It is not a beta lactase. It is a beta lactamase gene product. Mechanism: degradation of the ampicillin. Degradation of ampicillin. So, okay, so this is an important thing. Kanamycin. So gene product neomycin, phosphotransferase 2. Phosphotransferase, what is that? 2. This is very, very important. Okay. So what will be the mechanism? Covalent modification of kanamycins. Covalent modification of the kanamycins. Okay. Covalent bond is a weak bond, isn't it? It can be easily modified with the help of this gene. Tetracycline. Tetracycline is also another antibiotic. The gene product will be ribosomal production proteins. Ribosomal production protein. Protein already in our protein is essential for protein biosynthesis also takes place with the help of the ribosome only, isn't it? So the ribosomal production protein is very, very important. Okay, it's a gene product. A mechanism: efflux of tetracycline outside of the bacteria. Efflux. What is that? Efflux means, what is the meaning of that one? It may be elucidate out, I think, okay, of tetracycline outside the bacteria. And chloromopenicol. Okay, the antibiotics. Gene product chloromopenicol acetyltransferase. Acetyltransferase. Okay, this is important. What about the mechanism chloromophenicol to acetyl chloromophenicol? So normal one, when you are adding the acetyl, this enzyme is there, no? This enzyme act upon the chloromophenicol, you will get the byproduct, the mechanism, chloromophenicol is also to acetyl chloromophenicol, you will get it. You add acetyl from here to here, that's all. The help of the enzyme is called as chloromophenicol acetyl transferase, transform. Okay, it's also modified into some other form. So that is very, very good. These are the antibiotic resistant genes and their mode of mechanism. So these are the 4 things have been given. So maximum they ask the question ampicillin, kanamycin, tetracycline. These are, it's also chloromophenicol is also there. And because these are the important antibiotics mostly involved in the genetic engineering also. So, and also related to the plasmids. So it is very, very important. Okay. See the next one, the DNA transfer into the mammalian cells, culture in vitro. Culture in vitro means within the lab. Okay. Using non-viral vector. It's termed as a transfections. Okay. It's termed as a transfection. Don't forget, my dear student, what is my transfection? Sometimes they may ask the question, non-viral vector used for the mammalian cell culture. That is very, very important. DNA transfer into the mammalian cell culture in vitro using non-viral vector is termed as a transfection. Transformation is a procedure through which a piece of DNA is introduced into the host bacterium. What is by transformation? Transinformation. Okay. Transformation. What is my transformation? Gene transform. From where to where? Any one of them. Okay. The same gene is also we can transfer to the host of the bacterium. The gene transformation is called as what is that? Simply called as transformation. Okay. So procedure. Okay. Transformation is a procedure through which a piece of DNA, don't forget, this is the part. A piece of DNA is introduced into the host bacterium. What we did in the previous slide, we are collecting the different strains, plasmids in different sources and introduce into the host. Which one is the host? Here the host bacterium is which one, you know? Maximum. Already we discussed. Okay. Okay. Which one is the here? You know about the E. coli. Escherichia coli is the maximum used for the, okay. And also this is very, very important. E. coli. Don't forget, my dear. This is very, very important. So it is another important things. These are the things already we discussed. Now, what we discuss, you know, what is my marker gene, how it will be act, what are the types of marker gene, reporter gene and also selectable marker, how it will be act as a reporter gene, how it will be act as a selectable marker. Okay. And then what are the difference between selectable marker and also reporter marker, and then how it will be act. Okay. What is my non-essential region? What is my essential region? Okay. Non-transformation and transformation. Marker gene, what they will do. All those things already we discussed. And then antibiotic resistant gene and their mode of mechanism already we discussed. 4 antibiotics is there and corresponding gene product and as well as the mechanisms also already we did. Next, what is by transfection? What is by transformation? Transformation is nothing but the a piece of gene is introduced into the host bacterium. Is nothing but that process is called as what is a transformation. And then, okay, and then, okay, so and then the DNA is transferred into the mammalian cells culture into the vitro using non-viral vector is termed as the transfection. Okay, so these are the very, very important. So many times they were asked the question in the transfection. Transformation already know, but the gene is transferred from the one organism to another organism. That process is called as transformation. Mostly they were used the E. coli. Okay, go to the next one. Joining of DNA molecules. Joining DNA molecules. What is by joining? The enzyme used to join DNA fragments are called as DNA ligases. Already know, but so many times they were asked this part, isn't it? So many times already we studied this. DNA ligase is nothing but the one of the nitrogen fragments. Nitrogen made up of what is that? The protein molecules and also it's nothing but the one of the enzymes. Okay, DNA ligases. What is the meaning of this one? Ligases nothing but linkage, linking of the fragments. Those suppose take an examples of replication process, semi-conservative replication already studied. During that time, what happened, the 3' to 5' you will get that so many fragment is there, small fragment is that fragment is also called as, what is that? Ogasagi fragments, isn't it? The Ogasagi fragments also united with the help of the ligase enzyme. Then only you will get the complete sequence, complete double stranded structures, isn't it? Already studies that things. When the vectors DNA and foreign DNA are both cut with the same restriction enzyme, the overlapping end of the vector or foreign DNA are compatible and complementary. Okay. How it will be matched? Because about that one of the foreign DNA is also there, another one is also vector DNA is also. How it will be joined together? How it will be exactly fixed? Okay. With the help of the enzyme only. Already about restriction enzyme, it can be screening the which one nucleotide, where we have to make a cut. Okay, he analyzed that the polyandronic, already about the polyandromes sequences. He scan and also identify the position array and then cut automatically, already about. Same process also done by the both. Okay, whether it may be a vector or foreign DNA, both. Then only it will be exactly fixed. Otherwise, what happens? It will not fix and also overlap, and it will be some complicated is that. Okay, that is very, very important. These DNA fragments and vector molecules, when mixed together, form complementary base pair between overlapping terminal single standard DNA sequences. Okay, single standard. This is very, very important. Already we studied this sticky end, isn't it? Single standard sequences, nothing but the sticky ends. Both. Okay, donor as well as the vector molecule. The fragment is also done with the help of the restriction enzyme nucleus enzymes. So after that, what happens? There is a sticky end both. Then it will come and contact close together. As a result, it may be exactly fixed with the complementary genes. That is very, very important. This is the final step of the construction of a recombinant DNA molecule. After that, both A and B molecules exactly fixed, and then what happened? This process is called as ligation. Already we did that things also already we discussed. Okay, in the slide already what about the basic things. See here. So already we did this thing. Okay. See. So this is the A, and same process also repeated again and again. So I will draw a single diagram only. Okay, single line only. So this is also another one. Okay. When you are making cut, what happens? So the structure will be like this, isn't it? Already we discussed. Okay, this part. Okay, this part. So this is called a sticky end. This is the part is sticky end. Okay, likewise here also you will get two different structures. See here. Okay, one is like this, another one is like this. One is like this, another one is. This part is called a sticky end, isn't it? Sticky end. Here the single sequence is there. For example, adenine. Okay, here the guanine is there. Here also you can see that cytosine. Here adenine, whatever it may be, you can. Okay, depends upon. So this is the things. Okay, after the combination of these two means what is that? This gene exactly fixed with this gene. How it will be possible? This portion. So take an examples of this portion. See that one. In this portion, okay. How it will be exactly fix this gene. Okay. As a result, what will be the product? See that this process, how it will be get it? You will get these two sequences. See that one. This is the gene A. So this one. Okay, this is considered A. This is B. Okay. So this portion is here. This portion attached with the here. Okay, this portion. Take an example. This part. This part is also joined here. How it will be? See that the symbol start here. Okay. Likewise. Sorry. See that this one exactly fixed one, this one, correct only. Okay, so this portion, okay, this portion, imagine this portion, okay, this portion joined with the this portion, okay, this portion. See that one. Likewise, you will get it. Both you will get in this part. So here, what happened? You know, there is a sticky end is there in this part. It is not a join, isn't it? In this category, what happened? You know, there is one of the enzyme. That the enzyme is called as what is it? You know, a DNA ligase. The DNA, what is a ligase? The DNA, what is a ligase? DNA ligase, what happened? It will be linked together. Because both the gene, it will be unite together. So as a result, what happened? It will be completely joined. Okay, it is completely joined like this with the help of the enzyme ligase. So finally, what happened? You will get the structure. Okay, you will get the structure like this, isn't it? You will get the structure like this, isn't it? See that? I will get the DNA sequence. What is it? You know, like this, isn't it? So you will get the structure will be like this. The plasmid like yeah, both the character you will get it. So this phenomenon is called as what is it? You know, ligation. Ligation means what is a linking, ligase. Okay, ligase, DNA ligase. Okay, ligase. Ligase, yes, ligase. Okay, ligase nothing but linking of two genes. These two genes are united together, and also the cut is also made different. This one is also joined to this one. So we will get the two pair of genes. Okay, two different genes, two different combination of genes, like that you can tell. So that is very, very important, my dear students. Okay, don't forget, this is also important. See this one. See that this is the final step of the construction of the recombinant DNA molecule. The process is called as after the ligation. What happened? Okay, see that. See, the concept booster also is the one of the things. The preparation of DNA to be cloned. The DNA to be cloned can be obtained by cutting using restriction enzyme from genomic or organelle or DNA. Okay. After that, what happened? You know, it may be transfer into the PCR technology. What is that PCR technology? Polymerase chain reaction. You will get the more DNA molecule, exactly what you are going to clone. And then chemical synthesis also this. Okay, these are the process. And then passenger DNA, it is a DNA that is transferred from one organism into the another by combining it with vehicle DNA, complementary DNA, cDNA, synthetic DNA, sDNA. Okay, this is called as passenger DNA. It's nothing but used for the process. These are the two types of DNA have been selected. Okay, this is also very, very important. And then competent host. What is the meaning of competent host? Okay, see how it will be. When you are introducing the host, any one of the plasmids, sometimes it may be incorporated with the host DNA. Sometimes what happens, it will not incorporated with the host DNA. It will be present as it is. Suppose a plasmid is enter inside, what happens? It will not mingle with the host DNA. Okay, sometimes it may be present as it is. There is no any incorporation takes place within the host cell. See that type of things is called as competent host. Competent host is essential for the transmission with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through the cell membrane. In order to force bacteria to take up the plasmids, the bacteria cell must first be made competent to take up DNA. Okay, competent means it accepts the DNA molecule from the donor. Okay, that is very, very important. This is done by treating them with the specific concentration of divalent cations such as a calcium. This is very, very important, my dear students. So many times they were asked the question. Competent host is also treated with the help of a calcium ion, divalent calcium ion. With increase the efficiency, with which DNA enter the bacterium through in its cell wall. Okay, how it will be enter? Already we discussed these things also when you are introducing the calcium chloride, I think. Okay, CaCl2. So that time what happened? No, it also bind with the cell wall of the bacteria. During that time, what happened? It make a small pore like structure, so the penetration is very easy. Okay, that is the things here. Okay, see that one, then only it will be better for your understanding. So this is done by treating them with the specific concentration of the divalent cation, such as calcium, with increase the efficiency with which DNA enters. It can be easily enter the DNA molecule because it is a hydrophilic molecule. DNA is a hydrophilic molecule. It cannot pass through the cell membrane. After the treatment of the calcium, what happened? It can be easily entered. That is the reason. Okay, that's why we are treating with the divalent cation, otherwise called as calcium ion. Sometimes it is very, very, which elements used for the competitive cost for the DNA entering process. Okay, this is very, very important, my dear student. Meaning of competitive cost, the ability of the cost to perform certain task. Here it is referred to as the ability to bacterial cell to become host for foreign DNA. Competent host cell can be animal or plant cell to not necessarily need to be a bacteria. Okay, so competent means what is it? It's not only that plant cell. Okay, it is not only a animal cell. Sometimes it may be bacteria, sometimes it may be a plant, sometimes it may be a animals. Okay, so these are the important things. See that next one you can easily identify. See here in this category, this is the, what is that? Donor. This is the recipient. Okay, here what happened, you know, so it will not enter easily. It will not enter easily within the cell wall because hydrophobic molecules. Hydrophobic means it will not easily penetrate into the DNA molecule within the host. It will not allow. Okay. After the treating of calcium, what happened? The uptake of DNA by competent bacteria, what happened? It may be enter like this. Okay. It enter inside. Okay. After the entry, what happened? It may be split into two categories like this. It may gives a first one, how it will be, the result will be, it is incorporated with the DNA of the host. Here there is a separation of molecule. It can be easily incorporated within the specific site anywhere. Okay, this is the one methodology. Another one is how it will be, it is present as it is. When it enters, what happens? It will be formed as a separate molecule. Just it's nothing but the extra chromosomal body, nothing but the plasmid itself. See that one? DNA may be integrated into the genome. This one. DNA may be, the donor DNA may be integrated with the host DNA, right? Next one, what happened? The DNA may be integrated into the host genome, or exist as a plasmid. It will not incorporate. It's also a separately, what is it, functioning? Okay, such type of things is called as, what is it, competent genes. These two types of genes also present in it. Okay, go to the next one. The selection of recombinant, it is also very, very important. Recombinant. The insertional inactivations, insertional inactivation of antibiotic genes can be used for the selection of recombinant cells. Antibiotic gene is very, very important. A vector is chosen where the restriction sites are also available for cloning within the antibiotic genes. Here, the inactivation, insertional inactivation of the antibiotic genes. Here, the vector chosen where the restriction site are available for cloning with the antibiotic genes. Insertion of the foreign gene into the restriction site will lead to the loss of activity of the selectable marker. That is very, very important. Which side we are going to introduce? That is very, very important. Okay, so that is very, very important because insertion of the foreign gene to the restriction site. Restriction means availability. It will not allow. Okay, so as a result, what happened? The function, it will be incomplete. Okay, the insertion of the foreign gene into the restriction site will lead to the loss of activity of selectable marker. The selectable marker activity, already restriction means what is it? Antibiotic gene completely split the molecules. As a result, what happened? The marker did not properly act. As a result, it will be difficult. Then how will you insert? Restriction site. Other than restriction, we can use it. So then only that marker will be act. That is very, very important. See that one? For example, BP322 have a several restriction site. Already we studied. Okay, ampicillin. Okay, and BAM-H1 cut one position within gene that code for the tetracycline resistance. The recombinant BP322 carrying a foreign DNA at BAM-H1 site, listen here, BAM-H1 site, will not confer resistance to tetracycline, but are still resistant to ampicillin, which means elsewhere. This is very, very important. What they are doing, for example, when we take a plasmid BR, bubulin and rotary tube, 322 have several, a restriction site is also there. Particularly, BAM-H1 is a site. Okay, cut at a one position with the gene that code for tetracycline resistance. This gene is responsible for tetracycline resistant genes. Okay, so thus recombinant PP322 carrying a foreign DNA at this site will not confer resistance to tetracycline. It will not confer the resistance to tetracycline, but are still resistant to the ampicillin, which remaining elsewhere. That is very, very important. Okay, so don't forget. See that next one, you can easily understand. This is the ampicillin resistant gene. This is a BAM-H1. Tetracycline resistant gene is also ampicillin. The normal vector molecule. See that the recombinant PP322 molecules. See that new DNA is inserted in this portion. Okay, so this is the two differences. Insertional inactivation. Okay, insertional inactivation. Okay, how it will be? Already we discussed these things. Okay, so go to the next one. These recombinant cells are selected by replica plating method. How will you identify the recombinant cells? The recombinant cells are selected by which method? Replica plating method. That is very, very important. The transformed cells are first plated on ampicillin containing medium. Ampicillin containing medium. And after the selection of the transformed form, non-transformed, the colonies are replica plate on the medium containing tetracycline for screening of the recombinant clones. Here ampicillin, here tetracycline. Okay, so this is very, very important, my dear students. So today I think so we will discuss remaining things on the next classes. I will summarize all those things. Today, what are the things already we discussed? First of all, you should know what is my marker genes. Okay, what is my marker gene? Monitoring and detection of the plant transformation system in order to know whether the DNA has been successfully transferred into the recipient cell is done with the help of the set of genes, marker genes. The marker genes are introduced into the vector along with the target set genes. Okay, this is very, very important. See the marker genes already in our label. This is nothing but the plasmids. It has two things: is a target genes, and also which one is the interested one, and also selectable marker is also present in it. Next one is a markers also divided into two categories. What are they now? One is called as a reporter gene, another one is called a selectable marker. Okay, the selectable marker. Reporter gene already in our label. It is nothing but the reporter gene and screenable marker and also scoreable marker genes also. Okay. So it gives a available that shows the immediate expression in the cell tissues. A reporter gene is a test genes whose expression result in quantitatively phenotypically. Okay, that is very, very important, my dear. So the next one is that this is a gene already in our label. Okay, this is the marker gene, selectable marker. Okay, along with the gene interest. Selectable marker, selectable transform cell is a key factor in developing the successful method for the genetic transformations. This is done by certain selectable marker genes that are present in the vector along with the gene of interest. Already we know about. Selectable marker are an integral part of the plant transformation strategy. This is very, very important for the plant strategies, transformation strategies. Selectable marker genes enable the transformed cells to survive on media containing toxic level and of selection of agents, whereas non-transformed cells get killed. Okay, this is the mechanisms. Okay, and you know about the selectable marker versus a reporter marker. What are the general characteristics is there? What are the difference between this? What are the example is there? That is very, very important, my dear students. The antibody resistant gene and herbicide resistant gene is very, very important here. That is the GFP and also luciferase. Okay, and then So marker genes help in to identify and eliminating non-transformant and selectively permitting the growth of the transformant. In order that foreign DNA can be cloned into the plasmids, the plasmid should possess only one cleavage site for the at least one restriction enzyme, and this should be in a non-essential regions. Okay, that things also already we discussed. And also that what are the antibiotic gene product mechanism, ampicillin, kanamycin, tetracycline, chloramphenicol. What are the gene product is there? What are the mechanism is also there already we discussed. And then transformation transfection, that is very, very important. In vitro using non-viral vector used for the mammalian DNA transfer, that process is called transfection. Transformation is nothing but any piece of the, it's nothing but the procedure, any piece or any segment of the DNA molecule transfer into the host of the bacterium. Such type of process is called as, what is that? Transformation, DNA transformation or gene transformation. Joining a DNA molecule, already you know, any two DNA fragments are united together with the help of the enzyme is called as DNA ligase. Okay, it is very, very important. And the process is called as ligation process, DNA ligation, linking, and also DNA union, which was done by which one is the DNA ligase enzyme. That is very, very important, my dears. Don't forget, this is also a important thing. And preparing of DNA may be cloned, and passenger DNA is also very, very important. This is very, very important for the cloning methodology. Okay, and then competent host, you must note it down. This is calcium is very, very essential because the DNA molecule is a hydrophilic molecule. It will not enter easily into the cell membrane. Okay, the calcium, when you are adding the divalent cation, what happens automatically? The cell will easily allow the DNA molecule within the host. That is very, very important. Okay, my dear. So remaining things we will see the next classes, my dear students. Thank you so much for the participation. Thank you. Bye. Take care. Good night. Thank you, ma'am. Thank you so much.
Thank you.